Hi, Jacqueline.
Hi, Paul. How are you?
Good.
Hi, Nat. How are you?
So I guess
Hey, Jacqueline.
I know I'm master of ceremonies here, but I wonder if that means I'm supposed to decide when we start.
Yes, I think you're the boss here.
All right, okay. Well, listen, we'll give it 2 minutes. It's three exactly, just on general principle.
Perfect.
Paul, one thing when we go to start, you may need to assign me host unless you've already enabled screen sharing.
Okay. Let me do that right now then.
I think I'm the let me see.
Oh,
Yeah, maybe if you could make sure, put up your title slide, maybe.
we can do that. Let's see what happens.
For all those on the line, we're going to get started shortly. We just want to make sure
That work?
that's perfect.
Got it.
Then we are good to go.
All right, well, I guess we'll start. So I'm Paul Watkins. I'm one of the 4 PIs of the newest CERSI, the Triangle CERSI. And I understand it's usual at these webinars to present the results of a funded CERSI project, but since we don't have results, we're too new, we've chosen to present instead an exciting initiative that's occurring here in the Triangle that I think is facing some regulatory challenges that we hope will be solved by advancements in regulatory science. And it's my pleasure to introduce Nat Moorman, who's an associate professor at the School of Medicine here at University of North Carolina. And he earned his PhD from Wash U in St. Louis in molecular biology and microbial pathogenesis, did a postdoc at Princeton, and his own lab is studying the basic mechanisms by which viruses hijack cellular machinery to facilitate their replication. But of relevance to our talk today, he is co-founder and scientific advisor for READY, which stands for Rapidly Emerging Antiviral Drug Development Initiative. And the title of his presentation is Ready for the Next Pandemic. So Nat, take it away.
Thanks, Paul, and appreciate the invitation to be here and talk to you today. I'll go ahead and put my slides up. Sorry, we had a little. All right, can everyone?
Yep, looks good.
Excellent. Well, again, thank you all for having me here. Apologies for the delay at the beginning. Just want to briefly kind of set the stage for what I'm going to talk about today. So first of all, I want to sort of describe the problem that READY is trying to solve, which is basically preparing for the next pandemic by making antiviral drugs that work against families of viruses that cause pandemics. I want to share a little bit with you about the strategy that we're using, progress that we've made to date, and ongoing work. And then at the end is really the part that I was excited about to come talk to you today, and that's to ask for your help. We have obviously a number of challenges regarding the regulatory space, which I also think are opportunities, and finding those solutions to these challenges really could help stimulate desperately needed work in this area. So I'm hoping at the end of our talk, we'll have some time to engage in some conversation about ways we can overcome these challenges. But first, I'd like us to take just a second at the beginning, as we are hopefully on the back end of the COVID-19 pandemic, to really stop and think about what we all just lived through. The immense damage that's happened over the past 3 years. There were over 3 million lives lost to COVID-19 before vaccines were available. $14 trillion in damage to the U.S. economy alone. More deaths than all the combat deaths since the beginning of the United States. And the problem doesn't go away, as we expect there to be potentially up to 24 million cases of long COVID here in the US. So all of that happened because I think we would all agree we weren't really fully prepared. And so as you look at these numbers on the screen, I'd invite you to think about the impact that having medical countermeasures in place for viruses like SARS-CoV-2 really could have had. And the reason it's so important that we take that time to think about that is because, unfortunately, what we just saw wasn't really an unlikely event. And in fact, the odds of another event like COVID-19 have been increasing over time. Over the past 30 years, we've seen more and more new viruses continue to emerge. Some of them we knew about before. Many of them may not even have existed before they came to our attention. So this is a problem that we're going to continue to face, and that threat will become more and more pressing. Obviously, vaccines play a key role in how we deal with widespread viral outbreaks, epidemics, and pandemics. But at least with current technology and current approaches, vaccines are reactive. They require that a virus is circulating already, that we understand what that virus is before we can develop an effective vaccine. In that gap between the time when we can make that new vaccine, we truly need effective antiviral drugs to treat sick patients. And I think, unfortunately, another sad fact that we've learned over the past few years is even when vaccines are available, not everyone will take those vaccines or have access to them, which really highlights the need for antiviral drug development for these viral diseases. A question I get a lot when I talk to friends and family in particular is, what will be next, Nat? What will the next virus be? And the truthful answer is we don't know. When we look back at the past, at recent viral outbreaks and epidemics, most of those viruses were new to us, or viruses that we didn't consider to be major threats. Obviously SARS-CoV-2, but also think back to viruses like Zika virus, chikungunya virus, viruses that had explosive growth that spread across entire continents in a very short period of time. They were known, but they weren't considered to be widely circulating or imminent threats. So how do we know which virus to prepare for? Since we don't know which one comes next, what we do know is that the viruses that cause outbreaks, epidemics, and pandemics largely come from specific virus families. And so for those of you who don't think about viruses a lot, viruses cluster into families based upon their relatedness. And viruses within each of these families share common features that we can, in fact, begin targeting with antiviral drugs right now. For those drugs to be effective in the absence of a known threat, we need to plan for those drugs to work against an entire family of viruses. And that's what we call a broad spectrum antiviral drug. I want to define that term now because often when we say broad spectrum, people think about what we call a pan-viral drug, a drug that works against all viruses like a viral antibiotic. As much as I would love to discover and develop a pan-viral drug, I think that the probability is unlikely. However, there is quite a bit of evidence out there, including from existing antiviral drugs, that in fact the broad spectrum approach is very practical. Again, our focus at Ready is exclusively on small molecule drugs, and we'll get into some of the reasons behind that. So again, the antivirals that we're seeking to discover and develop work against an entire virus family, and that provides two main benefits. The first is the vast majority of existing viruses in all these families do not have approved vaccines or antiviral drugs. So a broad spectrum drug would provide treatments for existing viruses where there are no medicines. But at the same time, those same drugs would provide protection against future threats. The other focus of Ready in our drug discovery and development efforts is on orally bioavailable drugs. So drugs that can be delivered as a pill. The reason is that those pills can be shipped immediately without refrigeration. They're relatively stable, don't require cold chain storage. And so those drugs, if available, can reduce disease, hospitalization, and death, and in many cases can slow virus spread. So the goal at Ready is actually to develop 2 broad-spectrum drugs for each of these high-risk virus families. The other question I get a lot is, sure, Nat, that makes a ton of sense. Why isn't anyone doing this? Why wasn't this done before? Really, we've identified 3 major barriers. The first is what we call the discovery gap. So we can open up almost any journal and we can find academic research that's finding new molecules and testing new molecules in the early, sort of discovery and hit to lead stages of drug development. But again, those efforts are almost always about discovery, not on development. And that's because of a gap that we have in the way that we fund drug development, where most of the resources for this kind of research are available in the discovery phase. And there aren't nearly as many opportunities for funding in the academic world to move compounds from discovery through preclinical. The other gap we found is a gap in the pharma model. Businesses develop drugs to target specific indications for existing markets. So future tense isn't really a potential future threat, isn't a market that a pharma company can invest in. That's not to say anything negative about pharma, but it's a reality of the business models that they use. If I owned stock in a major pharmaceutical company, which I do not, I would be upset if I found out that that company was spending resources on a disease that may not ever even exist. And of course, this feeds straight into the disease X problem. There is no business model in place in pharma or elsewhere to fight diseases that don't yet exist. So really the challenge that we face is to incentivize antiviral development to prepare for the next pandemic disease. So we think that Ready is at least a major part of that solution. So Ready itself is in fact a 501c3 university associated entity that's acting in the public interest. So we're driven by our mission. And what we're trying to do at Ready is to accelerate broad spectrum small molecule discovery and development with a couple of goals in mind. And one is that we do have a firm commitment to equitable global access, which I'll touch on a little bit later. And of course, then addressing those market failures to disrupt that panic neglect cycle. For those of you who think about antiviral drugs, you've probably seen this or maybe even lived through it yourself, where in essence, nobody cares. A threat emerges, everybody cares. We put a huge amount of resources into finding a solution for a short amount of time. The threat recedes, and so does the funding to move the work forward. And that's why every time we're left with the gaps that we faced with COVID-19, because as I'm sure people on this call know much better than I, drug discovery and development takes much longer and much more effort than the typical duration of a viral outbreak. So I just briefly want to touch on the group of folks, some of the folks here that are helping to do this. So myself and my 2 co-founders, Ralph Barrick and Mark Heisey, have been working in antiviral discovery, development, and testing for several years now. The staff here at Ready, Jimmy Rosen as our CEO and director with Fletcher Ferry and Logan Ward. But the other thing I wanted to highlight is that we feel that to solve this problem, we need to bridge across more than just academic groups, but also into industry, government, and philanthropy. And so that's why we've recruited folks like Lydia Ogden from J&J, former Senator Richard Burr, Barbara Stevenson, John Bamforth, to help us as we work together to bring a global community to help solve this problem. So we're really, as I mentioned before, focused in 2 main areas. The first area is accelerating discovery. If we take coronaviruses out of the picture and instead we look across the virus families that are most likely to cause the next pandemic, what we find is that any existing assets largely reside in the hit to lead space. There are few, if any, drug candidates out there for viruses like Lassa virus, Nipah virus, or more broadly, arena viruses. So this is a role where Ready is helping bringing together groups to do that critical foundational research, taking a slightly different approach than most academic funding approaches, where we're really focused on project-driven, metric-driven, timeline-driven, high-risk research, which we obviously know will have a high failure rate, but accelerating that failure so we can find promising products in the early stages. But then on the opposite side, once we do find those promising initial assets, driving them forward through development. So that involves raising funds, be they government, philanthropy, or private funding, to de-risk those early stage assets, and then working together with innovators to collaboratively develop those drug candidates through phase 1. And so I think that I do want to take 1 second to discuss that. When we think about the next pandemic, if we could simply have these assets poised so that they're what we call phase 2 ready, imagine how much more rapidly we could respond when the next outbreak occurs. So I want to touch now briefly on some of the foundational principles of how we're doing this, and then talk about where we're at in our discovery and development process. So the first piece, as I mentioned before, is actually focusing on broad spectrum activity. So here I'm just showing you an example of one of our hit discovery and validation efforts. And this is, in this case, it's for the alphavirus NSP2 protein. So again, we use a multi-pronged discovery approach, where we're using, in this case, a DEL screen, which is supplemented by a biochemical diversity screen, a screen of a diversity library, and virtual screening, to find active compounds against the 2 functional domains of the alphavirus NSP2 protein. It's both a protease and a helicase, not uncommon for viral proteins to be multifunctional. And then really following a standard discovery process, with one exception. So while these initial discovery efforts focus on the chikungunya virus NSP2 protein, at all stages of discovery and into preclinical testing, we are always considering antiviral breadth. So if we find an active compound on the alphavirus chikungunya NSP2 protein, we immediately test it for breadth, activity against the NSP2 helicase and protease activities from Venezuelan equine encephalitis virus or equine encephalitis virus. We continue that testing throughout SAR driving rounds and medicinal chemistry. We continue breadth for antiviral testing in hit to lead, and also in preclinical development, using established animal models within our collaboration network to continue to ensure that breadth remains. And so any of you who've worked on an antiviral development program may know, if you focus on a virus, you will get a virus-specific compound. Keeping breadth in mind actually is a key decision driver as we move promising series forward. The other pieces I mentioned earlier is that we are very much committed to equity and global access. So an effective response to a pandemic requires global access to a therapeutic. The very word pandemic means global. So if we want to respond, we have to be able to respond, provide these drugs everywhere, and make sure that everyone can afford them. And I think one thing that gets overlooked when people talk about access and equity is that actually factors into the design of the small molecule inhibitors. For example, if we want to make sure that a drug that Ready develops is available everywhere, we need the cost of goods for a course of treatment to be $10 or less, $10 US or less. How do you make a drug that costs $10 or less? Well, you have to factor that into the compound itself. You need to be focusing and prioritizing drugs that are very easy to make. Simple chemical synthesis, typically 3 steps or less. The active pharmaceutical ingredient, or API, needs to be widely available. And so do the excipients that you might add into the formulation. And again, as I mentioned at the beginning, ensuring that the drugs that we make are orally bioavailable will make sure that we can distribute those drugs around the world where they're needed. The other thing that we're doing is thinking very seriously about the preclinical models that we're using in development. and I'll come back to this. This is one of the places where I need your help at the end, but I want to give you an example of the way we're approaching this. For many of these viral diseases, they're in fact poorly characterized. We have an understanding of the gross pathology, but we don't really understand the molecular events or all the clinical features associated with them. So we have a couple of projects underway now to deeply characterize human disease, but then go in and use genetically diverse mouse resources like the collaborative cross to find small animal models that accurately reflect both the pathology of the disease, but also the molecular features of disease in humans. We're using that to generate more closely aligned preclinical models. And in some cases, which I won't go into detail today, though happy to answer any questions, we've actually found that what we think is a disease, for example, severe COVID, is not a disease. While the pathology may be the same, in fact, the molecular features that underlie that pathology are quite different. So finding mouse models that accurately reflect those differences allows us to identify targets for new therapeutics, provides us with model systems to test them in, and also using associated EHR data from each of the human patients that we characterize allows us to find biomarkers and diagnostics to identify the patient population where we can effectively deploy the new therapeutics. So with that, those are some of the general principles and activities we're taking. And now I'd like to go into a little bit more detail as to what our pipeline at Ready currently looks like. So there's really 8 families of viruses that folks are concerned about when we think about pandemics. There's a lot of time spent talking about specific viruses and whether or not they should be on a list of priority pathogens. That is a useful and worthwhile conversation, but as I reminded you at the beginning, it's in some ways fighting the last war. It's the new virus which always sneaks up on us and causes the biggest problems. That being said, those viruses that are on the priority list don't have vaccines or therapeutics, so they also remain current threats. So hopefully this list of viruses is not too esoteric, and that you've at least seen some of these before. And right now I want to tell you about our efforts on 5 of these families in the next few slides. First of all, we're already doing some work on the paramyxoviruses.
But you see, I mean, if you look at this list, I mean, the list is always sorted alphabetically.
I'm sorry, let me go back. Sorted alphabetically, yes. This is not this is not in any order.
Like for instance, here it starts with number 2, not with number 1. Now, weirdly, they do somewhat weirdly, they do upscale now. They do superscript numbers, and that's the style.
I'm sorry.
I mean, I'm not doing this. The style, so that the system does it automatically, and I don't mind.
I think, Michael, would you mind muting for us? Thank you. Unmute.
Thank you,
All right,
everybody.
am I back?
Now we're good.
Okay, so I'm going to go ahead. I don't think that the comment about alphabetical order was necessarily about this. However, it was a good point. I believe they are in alphabetical order. And a good note, this shouldn't imply priority for one of these families over another. So the first family that we're tackling is the paramyxoviruses. So 2 example viruses in this family that are of particular concern and you may have heard of are Nipah virus and Hendra virus. So these viruses cause a severe influenza-like illness, but much more troublingly, they can progress to severe encephalitis with seizures and coma, including recurrent episodes of encephalitis that can occur up to months or even years after the initial infection event. These are very dangerous viruses, and we're very lucky they're not widely circulating. The case fatality ratio for infection with either Nipah or Hendra virus is between 40 and 70%. And as you can see, we don't currently have any approved small molecules, biologics, or vaccines. So looking across the field of Nipah and Hendra virus research, there's not much there. So we've decided to add our efforts right at the beginning. So on these viruses, we are currently discovering and validating new targets. In this case, we're actually focused on RNA targets for small molecule drugs. And we're working on finding new inhibitors of conserved RNA targets that would work on RNA structures that are conserved across the paramyxovirus family. The second virus family we're focused on, you might be more aware of. And so these are the filoviruses, viruses like Ebola virus and Marburg virus. So again, while disease may begin with a severe influenza-like illness that often progresses to GI symptoms that include abdominal pain, diarrhea, and vomiting, and then of course the symptoms for which filoviruses are unfortunately famous for: hemorrhage, bleeding, and bruising. In this case, we're slightly better prepared than we are for the paramyxoviruses. There are 2 monoclonal antibodies for one of the 5 Ebola virus strains. But again, no vaccines are currently available, nor small molecule drugs that are approved. Here we have multiple efforts underway, both in discovery and in hit optimization. So we're focusing on 2 viral targets right now: the viral helicase activity or NTPase activity and the viral polymerase. We're also performing small molecule cell-based screens with small molecule libraries to look for compounds with antiviral activity. I'm happy to report back from earlier this morning that we've been successful, and we do have active compounds that look like they have chemical properties consistent with further development. And so this slide is obviously just a little bit out of date by a few hours. Prior to this call, we do had a couple of hit compounds, just 2 or 3 from our target-based assays that we were moving forward in hit to lead optimization. Perhaps even more well-known than the filoviruses are the flaviviruses. So the flaviviruses include a number of viruses that both are endemic and cause widespread epidemics. You may be familiar with dengue virus, which infects 350 million people around the world each year, and West Nile virus, which is a significant concern here in the United States, or Zika virus, which emerged a few years ago and swept through South and Central America. This virus, again, causes, in these viruses, I should say, cause influenza-like illnesses, but really also severe muscle pain for viruses like dengue, encephalitis for viruses like West Nile virus, and birth defect for viruses like Zika. And more severe manifestations of each of those viruses can lead to hemorrhage. There are no approved biologics or small molecule drugs. There are vaccines for a few of these viruses. So of course, yellow fever virus has a vaccine that's been very effective, although we have seen recently episodes of vaccine breakthrough. Japanese encephalitis and tick-borne encephalitis virus also have vaccines, and there is a limited use vaccine available for dengue virus. However, we don't have vaccines for these other high priority flaviviruses like West Nile, Zika, and Powassan virus, for example, a virus that recently, in the recent past, unfortunately killed one of our United States senators here from the state of North Carolina, Kay Hagan. So there is an urgent need to provide treatments for these viruses. Here we have a lot of work going on. So we have broad spectrum target discovery and validation efforts for a number of different targets. We are developing assays for the viral protease. That's actually complete now and begun screening. We have hits coming out of our efforts for polymerase inhibitors. And we also have additional hits progressing from cell-based antiviral screens. A key partner actually in this program is Janssen. And some of you may be familiar with some of their very promising efforts, a very promising lead compound for dengue virus that we're helping them explore the breadth of its activity for other flavivirus indications. The coronaviruses, likely, unfortunately, need no introduction. Obviously, SARS-CoV-2 and SARS, but viruses like MERS, and of course, more common coronaviruses that sometimes get forgotten, like NL63, the cause of a common cold. This is caused by these viruses, flu-like illness, severe respiratory disease, sometimes with multi-organ involvement. And I think the area that's very concerning right now is, of course, the emergence of chronic disease or long COVID. So in the coronavirus space, it's different from the other families. In fact, there have been over 200 biologics developed with 2 deployed. Although, unfortunately, when we talk about these biologics, the monoclonal antibodies, none of them remain effective due to viral variation over time. There are several vaccines that are in use for SARS-CoV-2, but importantly, there are not vaccines that are effective against the other coronaviruses listed. There are small molecule antiviral drugs. However, they are currently only approved for use for SARS-CoV-2. So that's why we're actually still working on coronaviruses. Some people ask why focus on coronaviruses, but in terms of broad spectrum coronavirus drugs, that's really the focus of our efforts. So we are still looking for new chemical matter for a variety of viral targets. We are optimizing lead compounds, even on targets where there are existing therapeutics. For example, the main protease of SARS-CoV-2, Paxlovid, is a very effective treatment that targets that same target. However, Paxlovid doesn't have the full range of breadth against other concerning sarbecoviruses. And so we're working to identify derivatives that do in fact have that breadth. And we're also advancing a clinical candidate that targets the polymerase, even though remdesivir and molnupiravir target that polymerase. What we know and what we've already seen is that once a drug is widely used, especially a drug that targets the virus itself, we will develop resistance. The reason we're moving forward with this clinical candidate is because it has a distinct mechanism of action from either remdesivir or molnupiravir, and therefore may provide protection from viruses that evolve resistance to those drugs. Lastly, I'd also like to talk about our efforts for toga viruses. These are concerning viruses for several reasons. One, they do cause widespread outbreaks. Viruses like chikungunya virus, if you remember the introduction into the Western Hemisphere in late 2013 and 2014, which rapidly led to several million cases in the following months. But at the same time, viruses in the toga virus family have been weaponized, both in the past, both by the United States and by the Soviet Union. Viruses like Venezuelan equine encephalitis virus. So again, these viruses, like almost all of our human viruses of concern, can start with an influenza-like virus. But the viruses like chikungunya virus can cause a severe debilitating arthritis that can often become chronic. So 30 to 40% of symptomatic chikungunya virus patients go on to suffer from a chronic arthritis that can last anywhere from months to several years. And then viruses like Venezuelan equine encephalitis virus, as its name tells you, can cause a permanent brain damage, and encephalitis, and in many cases are lethal. At the moment, there are no biologics, vaccines, or small molecule antivirals that are approved, although there is a vaccine for chikungunya virus that is working its way through review right now. However, again, that vaccine is effective against chikungunya virus and not the other viruses in the toga virus family. Much like the other viruses, here we're moving forward. We're optimizing hit compounds on multiple targets. We have a promising lead compound that we're working on its stability, and we're also involved in efforts in preclinical studies for a potential clinical candidate that's targeting the viral polymerase. So altogether, this is what our prospective pipeline looks like of broad spectrum drugs for these virus families. As you look at this, what I want you to think about is the fact that to be prepared for the next pandemic, all of these bars need to be all the way to the right side of this graph. And until these bars are to the right side of this graph, we're not going to be fully prepared for that next outbreak. I'm really excited about this effort. Launching out of the COVID-19 pandemic, the G7 launched a group called the International Pandemic Preparedness Secretariat, or IPPS. And right now we're working very closely with these groups on one of the mission. So the 100-day mission is an effort to be able to fully respond to the next pandemic within the first 100 days. And we're helping develop the therapeutic roadmap for how we'll develop those therapeutics to be able to treat in the next 100 days. And I will say one of the things that we've been very adamant about is that 100 days is not how we prepare. 100 days is how we respond. And then in fact, it'll be a multi-year effort that leads us to be able to respond in those 100 days. I would be remiss if I didn't talk about our funders that are supporting this work. So READY is one of the AVID centers that has been funded by the NIH to move forward a lot of this work. But we've also been fortunate to have the support of the North Carolina legislature, the North Carolina Collaboratory, and the University of North Carolina at Chapel Hill in supporting these efforts. Our work on paramyxoviruses is moving forward through the PAD initiative. We're working on host-targeted antiviral drugs in collaboration with and with support from RTI International. The projects to develop preclinical models are being supported in part by SAS and the Collaboratory. And we've also received funding from the Defense Threat Reduction Agency for some of our efforts in the alphavirus, togavirus programs. So with that, I actually would like to transition to the part that I'm most excited about for today. And this is a conversation about some of the regulatory challenges and really opportunities that exist in this area. So drugs for these, for almost all of the individual viruses in these virus families would be first in class and first in indication. And as I've tried to reiterate throughout, and I'll say it again right here, for the vast majority of these viruses, there are no vaccines or therapeutics. Obviously, the same would be true for any newly emerging virus in these families. And one of the things that I wanted to point out that is the lack of a clear path to IND is a real challenge in discovery and development. You know, which virus strain should we use? Which preclinical models should we use? And how much preclinical data is enough to accomplish our goal of an IND and push these drugs through to the phase 2 ready stage? That has real implications that I'll talk about a little bit more on the next slide in the discovery phase. But this is where I learned a little bit of something about why we are so unprepared. What I've learned is that the unclear regulatory path is in fact a major barrier to investment. And the first time I learned this lesson is when we were talking to VC and private equity about supporting some of these existing assets. And this is a direct quote from a well-funded, well-recognized venture capitalist. He told me point blank, I believe you can make these drugs, but I'm not sure how you'll get them approved. That was a real wake-up moment for me. These are challenging markets, to say the least. And so I was surprised to hear that there was, in fact, private capital that was willing to step in at preclinical to push these compounds forward. But really, the regulatory uncertainty is really probably one of the major barriers to investor enthusiasm. So the question I would like to pose to this group, and I also want to preface this, I really hope that I have asked these questions out of ignorance and that in fact these answers are already out there. So please do not hesitate to let me know if I'm ignorant, because it would be a huge relief to have the answers to all of these questions. So how do we overcome the regulatory concerns about preclinical models for therapeutics? You know, is it possible to really define these models, the virus strain, the dose, the animals, the efficacy endpoints for each of these virus families, or at least for model viruses, preferably, in my opinion, 2 or 3 model viruses in each of these priority virus families. We've been doing quite a bit of looking here on the Ready side into the animal models qualification program, and looking for animal models that focused on these virus families that have gone through AMQP certification. I think that would really accomplish two things. So it would align us, folks out there trying to move these compounds forward, to align our discovery efforts to the expected IND. For example, when we're running our preclinical efficacy models, we'd be making sure that we are in a qualified animal model with the virus strain at the dose that has already been, has gone through review by the FDA. And that not only would it help the developers that are out there, but it would also help those investors to have some clarity and confidence in how we'll at least get through the IND process. Another question I have is can we, or how should we go about identifying and validating biomarkers for specific viral disease indications in advance? For example, viral arthritis or viral encephalitis. These aren't specific to a given virus, but rather to classes of alpha viruses, new world versus old world alpha viruses. Is there a biomarker we can use in a preclinical model that reflects what we expect to happen in humans? And then, of course, the other question, we want to position these drugs at phase 2 ready, so that we can immediately launch an efficacy trial on day 1 of the next pandemic. So in our thinking, our estimation anyways, to really be ready to launch that efficacy trial on day 1, we probably need more safety data than we would usually have, right? So there's no phase 2A, for example, which is safety in patients suffering from the disease, because the disease doesn't exist yet. So how much safety data should we generate in healthy volunteers with regards to number of patients, the dose range or exposure, and of course, do those studies need to be longer? How long should they be so that people have confidence launching into an efficacy study on day 1? And so just a few more questions for the group, and then I promise I'll be done. So, you know, when we start to think about that day 1 of the next pandemic, how do we design or pre-design a phase 2 study for an anticipated disease X indication today? So we don't know what the virus is, but again, as I mentioned, there are common features of these pathogens that medicines will be targeted at rectifying, right? Encephalitis, respiratory dysfunction. Can we use those features to think about the enrollment requirements and endpoints for those efficacy trials in advance? And can we think about what the various risk-benefit scenarios might be? We obviously have the benefit of hindsight for respiratory dysfunction with a virus like SARS-CoV-2. But imagine a virus like SARS-CoV-2 that had the lethality of MERS, which is around 30%. What does that risk-benefit scenario look like? How many patients would we have needed to enroll in an efficacy trial before we'd be willing to take that risk? I think these are questions we can think about in advance. We're not going to have concrete answers, but if we can at least have guidance, then we can think about how we're going to plan going forward and start to put at least the infrastructure in place. And that kind of leads to my next question. You know, adaptive clinical trials, I think, played an important role in our response to COVID-19. But back to these anticipated disease X indications, can we start to think about what adaptive clinical trials for these indications may look like for a future pandemic? Number of patients of group, maximum number of treatments to be considered. Again, all of that enables us to start to think about how we can position in advance with infrastructure, for example, clinical trial sites to test the different arms. So, you know, when we think about these viruses, you know, in general, disease X, at least historically, has not arisen in Canada. That's not the site, right? We know the sites where these virus families commonly circulate, and those are, again, likely sites for the next pandemic to emerge. Can we position clinical trial sites in advance and coordinate their efforts so that they have the infrastructure needed to immediately launch trials? And can we agree to in advance some standards for data collection and analysis around those predefined sites? And then, of course, an issue that I think would be incredibly helpful is, can we develop regulatory consensus for these emergency use events? Are there agreed upon standards with the specific risk benefit ratio that we can start to have that conversation now? And honestly, no matter how general those conversations are, the very act of having those conversations across regulatory agencies really sets that warm base for action when we need to deploy drugs that we hope to develop through READY. So with that, I'd just like to say thank you. And I'm hoping we can use the remainder of this time for you to tell me that the answers to all these questions are already out there and point me in the direction so I can go get that information. And with that, I'll go ahead and stop sharing.
Yeah, well, thanks, Nat, for a fascinating and somewhat scary, actually, presentation, waking us up. Obviously, great regulatory challenges there. If people are there, there's over 60 people watching. So if you can put your questions in the chat, I'll be happy to address them. But do you mind scrolling back to where you had all the viruses and sort of where you were, your current status slide?
Sure. 1 second. Let me get back to it and I'll pull it up.
Because I think the number I heard that you've sort of raised $66 million. Is that correct? Or committed? Has been committed?
Across all of our efforts to date, we've received a little over $130 million in direct.
What's that much? Well, this is my question. To really do this, are you talking about billions? I mean, there'd be no way to do it without major pharma, and is that what we're talking about? Is it that?
Yeah, that's what we're talking about. Although I do want to, so I don't want to scare people with the number. But on the interesting part, Paul, is that looking at this slide right here, for the vast majority of these viruses, the only things that exist are from lead to the left. And so interestingly, as we look across from discovery to phase 1, we need a lot of efforts to the left of lead. And those are some of the least expensive parts of this process. So we've done a full landscape analysis of what's out there for these 5 virus families, at least. And to get to the goal, based upon our efforts and other ongoing efforts, to get to the goal of 2 phase 2 ready candidates for each of these 5 virus families, we estimate that the amount is right around 1 to 1.2 billion. It's moving forward into the phase 2 and phase 3 trials and through to approval, where the cost exponentially increases.
Right. Yeah, for sure. We have one question. Can you share your slides? Is that okay? We can figure out how to post them. Okay. Well, so the other issue then, which I'm a little confused about, is that, there is the animal rule, the idea if you have a drug to treat fatal radiation, you can't do a clinical trial. So monkeys are accepted for efficacy and all you have to do is safety. And I guess you're talking about this, right? You're sort of saying, what is the animal model to use in the animal rule that would be accepted? Am I on the right track here?
Well, as I think about it, if the world, if developers knew that there were two animals, right, that we could test efficacy and safety in, that the FDA had already reviewed those models were standardized, they were well publicized for a specific virus, for example, then I've already got a goalpost. And I know with complete clarity for that virus where I need to develop towards. Now, obviously, there's going to be a conversation about whether the drug is efficacious enough, whether the benefit outweighs the risks based upon the safety profile. But at least I don't have to be concerned that the model I choose to do my preclinical work may or may not be acceptable. We recently helped survey existing preclinical models that are described in the literature and were available in development programs for a number of these viruses. There are differences in the models that are used. You know, which model's best, which virus strain is best for Lassa, which type of non-human primate is most appropriate in terms of the way it reflects them, recapitulate some aspects of disease, others, and it is very strain dependent. You know, but having that clarity that anyone who wants to make a small molecule drug or a therapeutic for Lassa, here are the models that have been reviewed and vetted. Here's the data that was provided to regulators in advance, potentially, as I noted on the slide, through the Animal Models Qualification Program. We've got clarity there as to what our expectation is. And that's, you know, models, as I understand it, that have gone through the AMPQ program, that is sort of the gold standard then for all efforts. That's a big de-risker for the investors and a you know, tells me exactly what I need to do for testing.
Okay, great. Other questions? People, please feel free to put them in, or as Nat has suggested, maybe clarification. So this is from Danica Pleku. Says, thank you for all this work and preparedness questions to the FDA. I was wondering, what does Reddy think about challenges regarding vaccine hesitancy and public trust for vaccine approvals and rollout, which we saw increasing during COVID-19? I ask this because, after all, vaccination as well as antiviral treatments are important factors during the epidemic. I guess what would be the implications for even a small molecule, which is your area of interest, with the distrust of, I'm not sure you're comfortable answering this, but even your own opinion. What do you think, Nat?
Yeah, I'll give you my opinion on this. And I think it's important. There's two things that I'd like to convey first. Well, really, I guess just one that's most relevant. So that piece is that I'm from Central Illinois. You can infer what you want from that statement, but let's just say when I go home and talk to friends and family, I am well versed in the issues that are raised around vaccine hesitancy and misinformation around therapeutics. Not to indict all of Central Illinois, if anybody else is from Central Illinois as well. But I actually think it's one of the really critically important things about an effort like Reddy. We're doing the work now. We intend to publicize the work that we do as we move forward. One of the most common concerns that I hear from friends and family is, but it was all so rushed. It just happened, and there was no time to see if these things were safe, and we don't know. Well, if we're telling that story consistently over time, and there's a record, a public record, and I don't mean a regulatory filing or a capital raise filing, but a record that people can search on Google or whatever the search engine is in the future, and they can see that no, they've been doing this over time. They've been making progress. At least those concerns about safety, they'll know that it wasn't a rushed process. You know, imagine if the story of remdesivir was widely known. You know, people thought with the EUA for remdesivir, wow, they just made this out of nowhere. That's problematic because then they think drugs get made in 3 months after an outbreak. But they also don't know about the decade of work that went into remdesivir that had it positioned for use in 2020. And that makes people nervous. So having that story out there, having that track record available on the internet for people to see, I think it helps to allay some of those concerns. Maybe I'm wrong.
Yeah. Okay, next question is, what's your perspective on screening diagnostic tests in parallel so that we can be early to intervene, I guess, by detecting the biomarkers you talk about earlier?
My perspective is it is absolutely essential. Diagnostics, vaccines, therapeutics. I think those are really the three pillars that I think about when I think about preparedness and response. A lot of people ask us, well, why aren't you working on vaccines and diagnostics? My answer is, I think therapeutics, just the small molecules, that's a big enough bite of the apple for us to try to tackle. But yeah, we absolutely have to have diagnostics in parallel. Recent chikungunya virus outbreak in Central America. Even if we had a drug that was effective against chikungunya virus, it may not be widely used. And that's because there are not available diagnostics in Paraguay and Ecuador to discriminate between chikungunya virus and dengue virus, right? So what is the right drug? I think in the event of another COVID-like pandemic, what I would love to see is a diagnostic that was available at home testing rapidly so that we could use a test to treat strategy where people who feel sick could test at home using a diagnostic, and they could drive to their local pharmacy and get where they'd have a prescription filled for a drug. I mean, that has huge implications in terms of spread, treatment, response time, all of this. So the need for diagnostics is absolutely critical. What would be fantastic is if we could have, I'll make this up on the spot, broad spectrum diagnostics. Knowing if it was an alpha virus versus a flavivirus in my chikungunya virus, dengue virus example. Just that level of knowledge, if we had an at-home test that said coronavirus accurately, and we know SARS-CoV-2 is widely circulating, I think that's probably a huge step forward in terms of how we respond.
We're nearing the end of the hour, but one intriguing idea is how the CERSI network could potentially, over the next 5 years, really contribute to this effort. And there's a recent RFA even out talking about collaborating with the CTSA network. I mean, there's few more noble causes than, protecting civilization. And I'm hoping this presentation leads to discussions of how we can really work with the FDA through the CERSIs, maybe this new potential collaboration with the CTSAs, to start thinking longer term of how we can really address this and be in the best position when the inevitable comes. I think
that would be fantastic.
Any closing words, Nat, before because it's been terrific, needless to say.
Well, I appreciate that, and thank you, and I appreciate you inviting me, and I appreciate all of you for coming to the talk. If people have the answers to those questions and you didn't feel like engaging today, please reach out. I'll be sure to put my email address on the slides. These are really big challenges for us. Some of the challenges we're facing right now, some of the challenges we're going to face as we develop these drugs. And so, our goal at Ready is to have the answers to these questions and develop them in collaboration with folks like the folks at CERSI and the folks at the FDA to put them in place ahead of time. Because we've got to be ready on day one next time. We can't do it again.
All right, thanks. Well, virtual applause. It was great. And, and we'll get the slides and distribute them to somehow, maybe through Tracy Chen or somehow to get it to anybody who wants them. I think you could also email Nat directly, and I'll make sure, or me, I'll have a copy. But thank you very much, and look forward to the next one of these. Bye now.
Thank you all.